Good morning or good afternoon, depending where you are. I'm Amber Salzman, and I can't tell you how excited I am to be facilitating this panel, having been involved with gene therapy and ALD for 20 years. To now have this illustrious panel and see how far we've come is a super exciting time. So we have Mandy Nagy from Mass General, and the fact that there is even a fellow who focuses on neurogenetics and gene therapy, time has come. We have Rich Calvin from Bluebird Bio, and it's exciting to see a product coming forward for cerebral ALD. And Eileen Sawyer from Swan Bio, where they're focusing on adrenomyeloneuropathy. So each of the panelists will present briefly background material, and then we'll break into question and answers. Please do post your questions in the chat. I'll be monitoring that, and we'll be questioning the panelists. So this is really important to get all the questions that I've been hearing offline with these experts on the call right now. All right, so let me turn it over to Dr. Nagy, and I believe you'll be projecting your slides. Wow, even more participants. I said high 50s, we're in the 70s. Just want to put that on the table.
Very good. Can you see my slides and can you hear me?
Perfectly.
Great. So thank you so much, Amber, for the introduction. Thank you everyone from ALD Connect for inviting me to give this overview of gene therapy this morning. So I'll start with my disclosures, which are that I have no disclosures to make. To briefly talk about the agenda, I'm going to give a very brief overview of genetics, just to give us a framework from which we can understand gene therapy a bit better. From there, we'll discuss two different approaches to gene therapy that are relevant for ALD. We'll move on from there to discuss some potential side effects that have been seen in these types of gene therapy, either directly in patients with ALD or in other studies, including patients with other disorders or in preclinical studies. And finally, we'll conclude by looking forward to all of the exciting opportunities and future directions within the field. So first, to understand gene therapy, we need to understand a little bit about the science behind genetic disease. What are genes? Genes themselves are small pieces of DNA found within each cell in the body. They're instructions that tell our body how to function. They contain information necessary to make all of the building blocks of our body, which allow our bodies to perform essential functions, things like creating organs, metabolizing the food we eat, and much, much more. So genes are small, but they're important in everything that our body does. Occasionally, there will be an error or a typo in these instructions, and we call that a gene mutation. So depending on the location of that typo, of what gene it affects, it can fundamentally change the way our body read and interpret these genetic instructions. While we see that the correct gene will make a typically functioning protein that will do any one of a number of functions, a typo or a mutated gene can either make abnormal protein that does not function at all, or in some cases can make no protein at all. Both of these can lead to serious consequences for the way all of the many complex systems of the body work together, can lead to a number of downstream effects, including diseases such as ALD. So as science has advanced and we've learned more about genes and what the instructions should look like under normal circumstances, the question arose, would it be possible to provide the correct instructions and to allow the body to produce a functional protein? Okay, so can we go from this mutated gene side back to the normal gene? And this is where gene therapy comes in. The goal of gene therapy is to provide the body with copies of the correct gene to allow for the production of appropriately functioning protein. The thought is to return the normally functioning protein to correct the systems that are out of balance and not functioning appropriately, and to treat or prevent disease that is present. The concept sounds very simple in and of itself, but the execution is quite complex, as you'll hear from myself and from the panelists who follow me. The first question being, how do we get this gene from the dish where we've created it, into the parts of the body that need it in order to function appropriately. And so the answer to this is also complex. There are multiple different modalities to gene therapy, but I want to briefly cover two that are relevant to our discussion of ALD. The first being ex vivo gene therapy, or gene therapy outside of the body or outside of the living. In this case, stem cells are taken from the patient. They are modified to carry the gene in question, and then they're introduced back into the patient. The other type of gene therapy is in vivo, or direct in the body, where the gene in question is delivered via a vector, which is often a virus like AAV, directly into the body to the tissues that require the gene. So to connect this back with ALD, we know that ALD is a genetic disease caused by a mutation in the ABCD1 gene. This gene is found on the X chromosome, causing it to present differently in men and women. We know that there are three different types of presentation. There's the cerebral form involving the brain, the spinal cord form, or AMN, and then there's adrenal insufficiency, or Addison's disease. Men, because they have one X chromosome, tend to present differently and can have any of these three forms or all three, whereas women typically will present with spinal cord disease only. So gene therapy for ALD, we first think of ex vivo gene therapy, which has been done for cerebral ALD, and you'll hear a bit more about. The idea here is as we had just discussed. So a patient will have stem cells removed. They will be transduced with the lenti-D vector, which carries the gene, the functional ABCD1 gene, and then those stem cells will be infused back into the patient. In order to make room for the stem cells, the patient will have to undergo conditioning or myeloablation to make room in the bone marrow for the cells to come in and to engraft. This has been studied extensively at this point with multiple years of follow-up. So just briefly, in previous studies, the initial study of ex vivo gene therapy for ALD, we looked at the neurologic function scale in patients who had undergone gene therapy. So the neurologic function scale, for those who are not aware, is a 25-point scale that looks at a number of different domains that are important in ALD, like hearing, communication, vision, feeding, walking, and incontinence. On this scale, a lower number is better. So 0 means that you're asymptomatic, and 25 means very incredibly symptomatic. And during the initial trial, it was found that 94% of patients, so 30 out of 32, had a stable neurologic function scale, meaning less than or equal to 4, and without a significant change, more than 3 from their baseline score after getting gene therapy. So these were good results. Now I want to move on briefly and talk about in vivo gene therapy. So in vivo gene therapy is different than ex vivo gene therapy because it's delivered locally within the body rather than outside of the body. This can lead to direct application to the central nervous system via injections directly into the brain, injections into the spinal fluid, or in some cases it can be given through the veins. Different genes are amenable to gene therapy in this manner. But the process here is somewhat similar in that you take the gene and it is conjugated to the viral vector. In many cases, that's AAV, or adeno-associated virus. The virus is able to enter the cell and essentially break down and leave the new gene in the nucleus, where the machinery of the cell is able to interpret the gene or the instructions and make the correct protein based on those instructions. As with any treatment, we have to think about potential side effects. And so I've listed a number of them here. And the good thing about knowing these side effects is that as we're gaining more knowledge, both from ALD and from other associated conditions receiving gene therapy, we start to understand what we need to look for and monitor for and how to treat these side effects. Certainly, in ex vivo gene therapy, you have side effects related to the conditioning or the chemotherapy, things like nausea, vomiting, infections, and fever. You can also have, primarily in vivo gene therapy, inflammation caused by the immune response to that viral vector. The immune response can affect things like the liver, which can increase liver enzyme levels. This is something that we're able to monitor and in most cases can mitigate by giving steroids to dampen that immune response. Related side effects include abnormalities of the blood cells, so things like myelodysplastic syndrome, which is a precancerous state in the blood, or leukemias or blood cancers, which have been reported in other conditions. Again, this is something that we are thinking deeply about, the causes of this and how to best mitigate this possibility. Other potential side effects, things like thrombotic microangiopathy, which is a very big term, as all medical terms are, essentially meaning blood vessel injury and small clot formation. Again, this is something that we're able to monitor for now that we know to look out for it. Monitor closely and detect it early, and there are treatments available if that occurs. And then finally, again, with in vivo gene therapy, there have been reports in animals, in other diseases, of dorsal root ganglion toxicity, which is damage to the spinal sensory neurons. Again, this has been seen in animals and has not been a significant problem in people. Despite those challenges, there are a number of opportunities available in the field. So adapting all of these different forms of gene therapy for various disease presentations that have been previously either had limited treatments or have been not treatable opens up a new realm for patients and families in ALD and also beyond. We're learning to anticipate side effects and develop monitoring and treatment plans, which is all part of the learning process, and we're making good improvements there. And finally, we're generating excitement around new therapeutic modalities and research into rare neurologic disorders. And as mentioned at the beginning, I am a neurogenetics and gene therapy fellow, the first at Mass General, with this new fellowship that was created by Dr. Eichler and my co-program director, Dr. Khurana at the Brigham. And I've included here just a number of the gene therapy trials that we have going on just at Mass General. And I think the creation of this fellowship really shows you how much the field is evolving and how much interest and excitement there is about developing these therapeutics and learning how to best treat patients. Thank you.
Great. That was a wonderful overview. Thank you. There's been a lot of interest, more participants, almost 100. A couple questions have already been posted. After we see the next two presentations, if they aren't answered as part of them, then I'll lead with those. But please, folks, keep posting the questions so that we have a great panel here, and let's take advantage of the time together. So with that, if we can move over to Rich, you're able to project.
Great, thanks, Amber. Is my screen sharing?
Yes, perfectly.
Excellent, thank you so much. And thank you all for allowing me to be here. I'm Richard Colvin. I'm the Chief Medical Officer of Bluebird Bio. And just make sure I can change slides now. I just want to disclose that one, I'm a full-time employee of Bluebird Bio. And secondly, that when I talk about Eli-cel, this is an investigational product still and not yet approved for use by the FDA. So, I really find it's a real privilege that we're invited to be here today, because I think, all of us face incredible challenges in our life, but the challenges that patients with ALD and their families face are obviously much more than most of us can even imagine. But what is amazing to me is every time I'm around the community or people who are affected, I'm always awed because, you know, the hope, the caring, and even the especially the love that we see is just so impressive. And so it's always our goal to be, you know, this is a kind of thing that drives us at Bluebird. It drives me for sure. And that, you know, for a long time, as Dr. Nagy's pointed out, there hasn't been anything for these types of genetic diseases. There hasn't been any treatments. There hasn't been any chance. And they don't affect as many patients, people as some of the other things. So it's hard for people to, especially companies that can make new therapies, to be able to get in them. So it's, to me, this is what drives me, though, is that the people like you and your families have been affected by something that we haven't been able to do very much about previously. And so, and that's what we do at Bluebird. So today I'm going to talk just a brief moment about Bluebird, a little bit of the approaches to gene therapy and our approach in particular, and what we have been doing in our clinical trials so far. I'm very fortunate. Dr. Nagy gave a beautiful slide of the data we have so far, and I think that's really important. And I'll touch on that briefly. But when you think about Bluebird, really what we're trying to do is develop curative gene therapies. We want to cure patients of their diseases. And we always talk about giving people more Bluebird days, but what does that mean? It means having a nice day with your family, being together, doing something you love, being able to have goals and think about the future. And so that's what our goal really is about. And so what we are focused on at Bluebird, especially now that we split the company into two, which you may have heard of, at Bluebird, we're focused on severe genetic diseases. And so at the moment, the ones that we have furthest along are sickle cell disease, beta thalassemia, and adrenoleukodystrophy. And because we've been around in this field for a long time, we've treated, you know, our programs and therapies have treated a lot of patients so far with gene therapy. We've had, you know, within our programs, we've had about 200 patients being treated. So we have a pretty good understanding of what these can do and what they cannot do at this point. And we're continuing on that pathway as we move forward. When you think of what we can do in the whole ex vivo or outside the body gene therapy, really it comes down to three different ways. And one is you could do gene therapy by getting somebody else's genes, right? So that would be an allogeneic transplant. That means somebody else is giving you their bone marrow, and then we replace your bone marrow with somebody else's bone marrow. And we know that's worked well for boys with cerebral adrenoleukodystrophy. The problem is not everybody has a match. And if you don't have a match who happens to be your sibling, sometimes the complications get greater. So there are two ways then to think about how you can fix those genes. One is by adding a gene, that's gene addition. Another way is gene editing, which you've been hearing a lot about. And gene editing means a lot of different things. And I mean, sometimes when people talk about it and they sometimes sort of think about it's correcting the mutation or the mistake, that typo that's in those genes. And sometimes that is the case, but other times gene editing can be gene addition. You can use gene editing to add a gene into a gene. So there's a lot of different ways to do it, and you have to pay attention to what people are talking about. What we have done at Bluebird Bio is gene addition therapy. And so we have three therapies that we're working on: Eli-cel for adrenoleukodystrophy, Beti-cel, which is for beta thalassemia, and BB1111, which is for sickle cell disease. And each one of these are unique in that they add back a gene that a patient with that condition needs. So for example, in Beti-cel, with Beti-cel for beta thalassemia, patients don't make hemoglobin. So we give back a part of the hemoglobin they need called beta globin. Same with sickle cell disease. It's actually the same protein that the patients need. So we give back beta globin. In the case of Eli-cel, we give back, make the patient's cells make this gene called ABCD1 that Dr. Nagy was mentioning before. So when it comes to Eli-cel, we have several clinical studies in ALD 102 and ALD 104. All the patients have been treated in those studies. And in those, we have 32 patients in ALD 102, which is what Dr. Nagy mentioned. And in ALD 104, we have 35 patients, again, all treated. And when patients finish the study, you may have noticed on her slide, there was a piece that said ALD or LTF 304 on there. And 304 is the long-term follow-up, because the studies 102 and 104 go for 2 years. And then after that, we end up following the patients for another 13 years, so that people are, the people who enter the study are followed for 15 years. And the way that these studies work is basically, the first thing we do is take out your bone marrow cells. And we don't take them out by pulling them out of your bone marrow or opening up your bone to get them. We actually just have the cells move out of your bone into your blood, and then we take them out of your blood. And then once the cells are out of your blood, they end up, we add that gene back. We add that in using the lentiviral vector that carries that gene into those cells. And once those cells now have that ABCD1 gene put in them, then they could start making that protein so that they could hopefully stop the progression of the disease. So the cells now are outside your body. So we got to get them back into your body. And the way we do that is by this process called conditioning, which is really chemotherapy to remove your old bone marrow and now put the bone marrow that's been, have that gene added back in. And once it's in, then you can actually end up with getting, starting to make that protein again. And we think what happens is that those bone marrow cells, some of them, they make all the different types of say, white blood cells and red blood cells. And some of the white blood cells we know go to the brain. We think, the way that we think this is working is that there's something called microglia. They're a type of white blood cells that move into the brain. And then once they're there, they produce that ABCD1 protein to help prevent the disease from continuing to move on. And the biggest thing is what again, like Dr. Nagy was talking about, is when during this whole process, the most common side effects are related to that chemotherapy piece, that conditioning piece. And because this is an autologous transplant, we don't have to worry as much about the infectious complications that, say, a transplantation from somebody else would cause, or we don't have to worry as much about any of the fact that we'd have to suppress your immune system so that that new immune system doesn't realize that you're not you, and it would start attacking it. So it makes it a little bit easier. This autologous transplant's a little bit easier than an allogeneic transplant. And I'll talk a little bit about this now. So what we do, so before I move on to this piece though, I will say that the way I think about how well LE-cel works is I think that for about 90% of the patients who get LE-cel, that they will stabilize and their ALD will stabilize where it is. And we've seen that for the long term. And we've treated enough patients to know we've treated some out to 5 years. And the way I think about it, 90% of the patients are going to do really well. And I think that that's pretty incredible, considering where we are. I'm sure you've heard in great detail about some of the safety events that occurred this year. Earlier this year, we had 2 boys who were diagnosed with myelodysplastic syndrome. So what is myelodysplastic syndrome? Really, what it is that it's sort of, back early days of medical school, the easy way to think about it is say it's a pre-leukemia. And at some point, those cells can become cancer cells and you get leukemia. It's not as simple as that. And sometimes that happens, sometimes it doesn't. Sometimes it causes other problems, and sometimes you don't make enough white blood cells. But what it means is that there's an abnormal cell that's in your bone marrow that's supplying a lot of the cells that are in there. And this is always the kind of side effect that we worry about when it comes to gene therapy, because we're manipulating the genes, and some of those genes, of course, are in your bone marrow, are really important for controlling how well cells grow and divide. And because of that, if you somehow interfere with one of those genes, either one that turns on growth or one that turns off growth, you can have an impact on those cells and as they might become cancer. So the good news though is that the 2 boys are stable now, and I think that's really important to remember. And the other is that we do think that this was caused directly by that that lentiviral vector integrating near a gene that could be involved in myelodysplastic syndrome. There's obviously more to it than that, and we don't understand all the other factors that are involved, and we do, but it is something that is unique to each of these patients. And the other piece of this, I would say, is that, you know, when you look at in the big picture, we've treated 60 something patients with Eli-cel, and these are the two who've had myelodysplastic syndrome. And at the moment, you know, there's no reason to think anybody else will progress, but we're keeping a very close eye on that, obviously. And so, but this is what we're working on. And because of these two cases, the program is on clinical hold right now, meaning that the clinical trials are not progressing. It's interesting because we don't have any more patients for those clinical studies anyway, because the studies are completely filled. But at the same time, we're working very closely with the FDA to get the studies off hold and to get Eli-cel approved in the U.S. The important thing here is that FDA is working very hard to make sure that we're doing whatever we can to keep the patients safe. And I think they're doing a great job of going into it. We're helping to try to understand this as much as we can. But this is going to be the importance of every gene therapy, every gene addition therapy, every gene editing company that we need to think about, that we're likely to see some changes in the genome that occur over time because of the therapies we use. And that's going to be how we move forward. So I'll be happy at the end to answer any questions about these cases or any others about this. But I do think that I feel very privileged to be here, and I'm thankful that you're giving me the opportunity to talk today.
Great, thank you. Should we move on? These are wonderful information. I already have seen questions, but we'll go through the presentations. So if we can now move on to Eileen, this is great. I mean, there's clearly a lot of interest in this topic.
Right. Are you able to see my slides?
Yes.
All right, perfect. Well, first of all, thank you to the organizers. I'm really, really pleased and honored to have the opportunity to come here today to share a little bit about what Swan Bio is working on. My name is Eileen Sawyer, and I'm the lead for medical affairs at Swan Bio. So that is also my disclosure that I am an employee of Swan Biotherapeutics. So a little bit about Swan Bio for folks who may not be familiar with us. We're a small biotech that was founded in 2018, so about 3 years ago, by two experts in ALD and AMN. One is a neurologist with deep experience, and the other is one of the leading patient advocates. And they came together with a scientist who has 25 years of experience in gene therapy to create the company. And they are infusing our company with the passion that they had, and that indeed each of us has, about bringing life-changing treatments to people who have genetic neurological disorders, focusing first on disorders of the spinal cord. And the company right now, as I said, we're small. We have offices up in the Boston area where I'm located, and the headquarters in Philadelphia. At the moment, we are focused on adeno-associated viral vector, or AAV-based gene therapies that Dr. Nagy did a very nice job of giving a brief overview of. Our lead program, which I'll talk a little bit about today, is SBT101, and it's in preclinical development for the treatment of adrenomyeloneuropathy. So that means that we're still working in animal studies. We're not yet conducting any clinical trials for this in humans, but we are working towards that. And one of the things that we're hoping as we work on this, one of the key problems in gene therapy, as Dr. Nagy said, is getting the genes to the right cells that need it. And in this case for AMN, that's in the spinal cord. So one of the problems that we're hoping to solve is how to get genes into the spinal cord in an efficient way. And when we solve that, we're hoping to be able to apply those learnings as well to bringing forward other gene therapies for other similar disorders of the spinal cord. But right now our focus is primarily on adrenomyeloneuropathy and our program SBT101. So I'm not going to spend a lot of time on this because the two panelists before me covered this, I think, very well. So there are two types of gene therapy. There's the ex vivo or cell-based delivery that Dr. Colvin was describing previously. That's shown on the left side of the slide here in gray, and involves taking the cells from the patient, modifying them outside the body, and then giving them back. And then there's the type that Swan Bio is focused on, which is shown here on the purple side of the slide, which is the direct delivery using a vector such as an AAV to directly infuse the gene therapy into the patient, where the vector can then find the target cells and modify them in the body directly. And this is more suited for something like the spinal cord, where we can't really take the cells out. We have to reach them where they are in the body. So although Swan Bio is working on the first AAV-based gene therapy for AMN, this is, the AAVs have been studied in clinical trials in multiple diseases for over 20 years now. So there have been over 3,000 patients treated in various clinical trials in various diseases using this class of medications. And this has really increased in the last 5 or so years, especially, as you can see in the graph. And in fact, as more and more work has focused on this type of gene therapy, 2 treatments have recently even achieved FDA approval. One of those is Zolgensma, which you may be familiar with. It's an AAV9-based gene therapy for young children with spinal muscular atrophy. The other one is called Luxturna. It was approved in 2017. It's an AAV2 gene therapy for a type of inherited blindness. And there are multiple others in late-stage development as well. So there is more and more experience with this overall type of gene therapy out there. So what exactly is an AAV gene therapy? So essentially what it is small particles that are designed to deliver the gene of interest to the tissue, the target tissue in the body. So there are 2 key components. One is this outside delivery vehicle that's called often the capsid or the protein shell. You may hear it referred to that way. And it's essentially just that. You can think of it almost like the cardboard box that a package arrives in. So it gets delivered to the right location in the body. The cell will unpack it, throw out that cardboard shell, and keep the contents. And what the contents are is called the gene cassette. And that's the piece of genetic material or DNA that contains the therapeutic gene that will produce the protein that we think will help the disorder. And the gene cassette contains both that therapeutic gene as well as some other elements that help the body correctly read and produce the protein. And Dr. Nagy went over this a bit as well, but I wanted to just briefly revisit it. So when that AAV vector arrives at the cell, it binds to the cell and gets taken inside, where the cell will unpack it, take off that outer capsid, and keep the gene material from the inside. And that will be brought into the nucleus, which is where all the DNA in the cell is stored. And once in there, it doesn't integrate into the DNA that's already there the way that some of the other types of gene therapy do. Instead, it forms what's called an episome, which is a stable little circle of DNA that sits outside the other DNA. And the cell will be able to read that and use it to make protein just as it would normally. So this is an important feature that that DNA doesn't integrate. And it makes it AAV gene therapy particularly well suited for targeting cells like the cells in the nervous system that don't divide or change a lot. It means that you can give one delivery and they'll have that gene for a long time, or at least so we hope. So what is SBT101? It is one of these AAV-based gene therapies. The outer capsid is what's called an AAV9. So that's a particular variant of the AAV family that is known to be good at transducing cells in the nervous system. And it contains the human ABCD1 gene, which is the gene that is affected in ALD patients. And we've designed this to be suited for intrathecal administration. So it will be given through a lumbar puncture into the cerebral spinal fluid that bathes the spine so that it can have direct access to the cells in the spinal cord that are affected in AMN. As I mentioned, we're in the middle of studying this in animal models. So shown here is some data that we've generated using a mouse model of adrenomyeloneuropathy. On the left side, we've given these mice the intrathecal infusion of SBT101, and then we've looked to see, are we getting to the cells we need to get to? Are they producing protein? What's happening? So in these mice, you can see on the left, we were actually able to detect that we were able to establish expression of the human ABCD1 protein in their spine specifically. So that showed us that we were getting into the cells, that whole process of entering cells, unpackaging, and then using the cellular machinery to produce protein was happening. And then if you look at the graph on the right, this is VLCFA levels, and this is, of course, the very important metabolite in AMN and ALD. And you can see at the higher doses, there's actually a significant decrease in the levels in the mouse spine. And so this tells us that one, the protein is being produced correctly, it's functional, and we're actually able to produce it at levels that are impacting a disease biomarker here. So this was all very encouraging to us. So together, those results really showed us that we are able to target the spinal cord, get SBT101 into the neurons we needed to in the mouse, and that we were able to do so at a level that improved markers of the disease. So we are now continuing to study SBT101 in additional animal models and animal studies, and working as fast as we can towards initiating clinical trials in the near future. In addition, we've started a natural history study of men with AMN in order to better understand the disease progression and help us select what some relevant endpoints might be so that we can detect early signals for safety and efficacy as quickly as possible once we are able to start clinical trials. If you're interested in learning more about that study, you can visit clinicaltrials.gov and search the NCT number that's on the slide, or contact the email address that's shown there for more info. Or I'd be happy to answer any questions in the Q&A.
Well, thank you all for wonderful presentations. We have a lot of questions, so let's try to be very focused and direct on them. Only because you just finished, Eileen, I'll take actually the last question since it's so relevant. Can you give any sense of the timeline of when you're going to initiate the clinical trial for AMN?
Yeah, we are in what's called IND enabling studies now, looking at establishing safety and toxicity and selecting a dose. And we are working as quickly as possible towards initiating those trials. I can't be more specific than that, but you know, as soon as we have news to share, we will be sure to make sure that that gets out to the patient community.
Okay, so let me go through, there's a lot of questions and we'll deal with more coming in. So the first couple were around who are candidates for each of these, and it had to do with young versus old. So let me just, for clarity, probably divide it into the different phenotypes. So are any of these gene therapy treatments available for young patients with cerebral ALD versus older patients? And the same thing for AMN. So I guess, Rich, maybe you can address for the cerebral ALD, who are your patient population now and in the future?
Yeah, great. That's a great question. So for cerebral ALD, the oldest patients we have are under 18. So we have had patients who are 17 years old, and anybody under that. Adult ALD is a slightly different indication, as you know, and we are interested in that as time goes on. In terms of younger patients, as we have more screening, we're going to identify boys younger, right? So I think our goal, of course, is to actually include very young children. Age 4 is not a problem, right? So when we have had some really pretty young children there. When they get below age 3, it becomes a little hard to do the apheresis piece of it and take their blood cells, get enough cells out or their bone marrow cells. But I think we're finding ways to do that now again, and the diagnosis takes time as well. So effectively, the way we see it, it's sort of around age 3 is where it becomes practical. that may change, but again, it might not be something we'd want to do for a child below 3. We don't think that there would be we'd be wanting to wait to see what happens before we started embarking on the treatment. But that's something that we're continuing to think about.
Great. So if I can throw out as a follow-up, since the practicality of under 3 is the apheresis, if the parents had stored cord blood, would you leverage that at all, or is that too experimental for you guys?
Well, that's a great question. I mean, that's again something we have thought a little bit, but we don't have. When you think about when you're trying to do a sort of to put it in, there's not an easier way to put it, but as a commercial way of doing this, when our manufacturing has to be so, there's such quality control on it, and it has to be so similar. We don't have that as part of it. That would have to be another experiment, and we'd have to try to figure out a way to get that done.
Great. All right, so I'm going to go through the questions in order. Some of these I don't think we can address here, because one of the questions was, are there other meds for ALD patients besides corticosteroids for the adrenal insufficiency? And right now they're experimental, and there's some palliative treatment. I am not a physician treating patients with ALD, so I do encourage you to talk to a physician about it in terms of what the standard of care is. Oh, the other one is a really good question as well, is what about we've talked about patients with cerebral ALD going in early. We've talked about adrenal mononephropathy. What about the unfortunate patients who get diagnosed and at that time they have a LESS score of greater than 10? I don't know, Rich or Eileen, if you want to say whether you're going to address that. Otherwise, I know there's other activities going on. Do either of you want to take that on?
I will take it. I mean, I wish there was an easy answer for that right now. I mean, that's, patients who've progressed to that level, it becomes very challenging. And, what we've seen in allotransplantation is that as the LESS score has gone up into that level, I should say, the outcomes haven't been, unfortunately, very good. I mean, I think we need to come up with new ideas, new therapies at that point. When I think of what gene therapy can do, and similar to allotransplantation, and it stabilizes as much as anything else rather than reverses. I don't know if Dr. Nagy wants to chime in on that either, because it's unfortunate that I just don't know of any way to address it at this point.
Yeah, if you don't mind addressing that, Dr. Nagy.
Yeah, absolutely. So, I think it is a tricky situation where we know that at those higher LESS scores, that the risk versus benefit of gene therapy at this point is tilted towards the risk side. And these patients, unfortunately, we haven't seen that they do as well. And so it's not something at this point it's safe. Hopefully in the future, we'll be able to advance research to that point. At the moment, our group is doing a lot of work looking into not gene therapy directly, but into quality of life outcome measures and trying to determine what things can we best address even beyond these gene therapy trials to improve quality of life in those patients, and hopefully, in the future move more towards expanding these therapeutic options.
Right. And it seems like there has to be almost a radically different approach than the ones we're talking about today. But there's other approaches besides gene therapy, because this is a really untreated population. The other ones, as not a surprise, Rich, can you address anything on two things? One, timeline of coming off hold, which I guess will impact when you would expect to have the engagement with the FDA on market authorization.
Yeah, no, great. They're great questions. So we're in ongoing discussions. It's not even like it's a when do we expect to hear from them? The discussions are ongoing. They are very careful. And I think, this is the precedent for this is really important, because this is going to affect all gene therapies in the future, whether it's for ALD or for any other diseases that anybody with gene therapy, gene editing is going to approach. I mean, you can see how they're very interested in AAV gene therapies, where they've had a big summit very recently on that. And really we had a long delay in sort of gene therapy. 20 years ago, it was the first golden age of gene therapy. And I think we're now in the second, but in the first golden age of gene therapy, it basically stopped because of the risk for cancer and leukemia. And of course, the Jesse Geisinger situation where this poor young man got gene therapy and then had a massive inflammatory reaction. And so they don't want to see anything like that happen again. Now, I think that that's less likely. We've, in the Bluebird studies, we've treated about 200 patients so far, and overall with lentiviral vector gene therapies, there's been about 300 patients, and of those, we have these two are the ones who've had myelodysplastic syndrome. What we know they don't want to see is that this is the tip of the iceberg, so to speak. Based on everything we've looked at, we have ways to study this in ways that we weren't able to 20 years ago. We don't think that's what's happening. We think that there's something unique about these two patients. Is it possible that this is going to happen again? I think it will, whether it's in LVV gene therapies like we have, or whether it's some of the other kinds of gene therapies. I think that when we do something to the genome, we're likely to see changes that occur in this way. So I think the key is to as Dr. Nagy pointed out, it's all about the benefit risk of this. And if the risk is that you have 2 out of 60 patients with cerebral adrenal leukodystrophy who end up getting myelodysplastic syndrome, when otherwise the disease would have progressed, otherwise the alternative, if they didn't have an alternative, I think that it's how you have to balance that out very carefully. So the next part of that question was, what about the marketing authorization, the BLA as it's called in the US? We're actually working on that at the same time and discussing that with the FDA at the same time. So when that gets filed, you will know as well. But we answer the whole idea is it's a very collaborative relationship with the agency, and we're doing our best to move it ahead, not only quickly, but safely for as many patients as we can.
Awesome. There was a comment related to this that they said there was some chatter on social media that you may be monitoring other ALD kids who were on the trial for myelodysplastic syndrome.
Yeah, no, good question. So the first thing is we monitor every boy on the trial for myelodysplastic syndrome. It doesn't matter if there's any hints or none of it. That's the biggest reason why we have the 13-year long-term follow-up study following the 2-year therapy. And the idea is because these kind of therapies are very new, and two, the risk is to see that you could have myelodysplastic syndrome or leukemia from them, and those things take time to develop. So we're always monitoring all the patients for that. We have some, the best way to monitor it remains simple blood tests that we do for patients as they go along, but we have some deeper ways to do it to try to understand how the vector has inserted itself into the cells. So the short answer is we monitor everybody for it, no matter what.
And there was one follow-on in terms of how close after the treatment did you start to see a signal in the blood?
Yeah, so in these two boys, both of them were diagnosed with MDS a little more than a year after they were treated, which again is different. We've had a lot of patients who have much more follow-up than that. So we do think and we don't understand what those differences are yet, other than the fact that they ended up with myelodysplastic syndrome, and we understand what we think is part of the cause of it is the lentiviral vector inserting near a gene that could be associated with MDS. And before that time, there were some post-treatment differences that we saw in their blood counts compared to some of the others. So it started right at the beginning in that way, but it was not myelodysplastic syndrome at that time. So again, it was something that we were able, that if it was somewhere else, we'd be able to see it probably in some other patients. We wouldn't know if it was myelodysplastic syndrome yet, but there was something there.
Okay. There's more questions we'll get back to, but first let's move on to AMN for a bit. Eileen, do you want to address the question about would you consider adding women to your AMN study either now or timeline on that? And how are you thinking about addressing the fact that they suffer as well?
Yeah, that's a great question. So we're starting out focused on men with AMN, in part because that's one of the areas that we have the most information about the disease. and that we think it'll be probably easiest to start to demonstrate whether or not there's an impact on the course of the disease. To truly be successful there, we would certainly be interested in considering women in the future. I think one of the things that will be challenging is that we do need to know more about how the disease progresses in women specifically, and what the major symptoms and complaints are in order to be able to appropriately design trials. So there's definitely a need to collect more information on what that looks like for women.
And are you progressing a natural history study so that you can address that question?
Yeah, so right now the natural history study is enrolling men, but as we get down the line, it is something that could be expanded to include broader patient populations.
Do you have any rough timelines of when you'll start to think about women?
I think we'd want to see some information from our first in human trials to make sure that there's some possibility of safety and efficacy.
I know we only have 5 minutes left. There were a lot of questions posted. There's a few critical ones where there's no, I don't think, answer. It's just more opinion. When do we think, I mean, on the assumption that we'll get past some of the challenges right now, and there'll be some understanding of the risk benefit for MDS, et cetera, and I don't think there's a magic to it, there's discussion. When do we think we would actually offer it preemptively? So given that autologous is safer than allogeneic, you're not, you know, the GVHD, all of that. When do we think we'll get to the point that you don't have to wait for a lesion and see if things get out of control, but you're born with ALD, when you're stable, before you start getting any kind of lesions, you go forward? And I don't know if Dr. Nagy, you have a view, and Rich, you have a view in terms of how we can be more prophylactic about when kids are diagnosed.
Sure. Yeah, so, you know, I think, again, this is a question of risks versus benefits. And we know that, you know, out of babies who are found to have the ABCD1 mutation, past data has shown about 40% developing cerebral ALD. But that's a large population who don't over time. And, you know, with the side effects and risks that come with gene therapy, you know, that we've discussed, I think, extensively today, it's a difficult decision to treat prophylactically when you know that some population of children are never going to need that therapy and are being exposed to these risks unnecessarily. So I don't know that that's what we're looking at right now, but I don't know if you have a different opinion.
I hate to cut it short. It's just we have like 3 minutes and there's still a few key questions. And I know you guys are, I could sit and talk all day to you guys. And I know that there are questions would come up. Eileen, there was one question which is very relevant. Can somebody enter the natural history study?
And that's a great question. So at the end of the slide, there was a clinicaltrials.gov number. You can search for it there, and there's information on how to contact the sites, or you can contact the clinicaltrials@swanbiotherapeutics.com email address, and we can help connect you with one of the recruiting sites.
And then great. And then one more question about, which I think we're being optimistic, and I know I am, we've come so far, is if you are diagnosed at birth, you're monitored, you get into the Lenti approach to prevent the cerebral from cascading, and now you're an adult and you don't want to have the adult form of the disease, AMN, could we then do an AMN treatment? I don't know, Dr. Nagy, if you want to hit that one as sitting in the middle of, you know, when do you think that'll be on the table?
I do not have an answer to that. So I think I'm not going to delve there. I think there's a lot to just move forward in terms of the AMN research before we even got to a question like that. I do want to go back to the previous question just for 1 second. And I think Katie Becker brings up a good point that, you know, despite the risks that I was talking about with prophylactically treating, we don't even know that the treatments prevent the cerebral disease. So, you know, we think that the open blood-brain barrier is part of the way that the gene therapy works. So even if we decided it were helpful, it may not be helpful to give this treatment prophylactically.
Okay, I'm so sorry that we couldn't address all the questions. Clearly, we have to have a special day on this and a learning session. You guys have been amazing in addressing the questions. You're a fund of information, and we're so pleased that you're driven to take this on behalf of patients and their families. So appreciate that and appreciate all the questions. Thanks.